Hello, everyone. I am Liz Sorani. I'm the Director of Research at FARA. Welcome to the FARA Flash Talks. This is the 6th year for this event, and it's part of our May celebration of FA awareness. Every year, we invite several junior investigators funded by FARA to present their research to our community. And so today you'll hear from grad students. These are researchers that are still completing their PhD studies, postdocs who are researchers in training after they completed their PhD, and also early career clinician scientists who are finishing up their residency. As you probably by now are familiar with the format of this webinar, we ask presenters to summarize their research in just 5 minutes and with just one slide, and to explain to us what they've discovered and what they plan to do, and also how this research can get treatments, to get us to treatments for FA. So let's take the opportunity to learn about FA research, but most importantly, to interact with these young investigators and ask them questions. So please type your questions in the Q&A box. We will get through as many as we can, and try to use the Q&A box and not the chat box if possible. Also, a reminder that we are recording these presentations and we will post them on YouTube. This is what you'll be hearing today. We'll learn ways in which FA affects the brain and the heart, and these are the two organs most affected in FA, and how we can use these learnings to develop therapies. Federica will tell us about what happens in the cerebellum, the part of the brain that controls movement, but also is important for other functions like speech, and how FA disrupts connections within the cerebellum that are important for this finely tuned movement coordination. Francisco will tell us what happens in the heart, where something similar seems to happen, and the heart rhythm goes off beat, and this causes arrhythmia when the heart rhythm is either too fast or too slow, or it becomes irregular. And then the last two talks will focus on two special cells called microglia that functions like immune cells, that is, as a defense for the brain. And these cells not only protect the brain against pathogens, but they also help clear out debris and support the function of the neurons, the brain cells. And Carla will tell you how microglia in FA becomes harmful for the neurons instead of being beneficial. And Yu Min will tell you how he's using this information to develop a treatment for FA, where he uses bone marrow transplantation as a source of these healthy microglia to improve the symptoms of an FA mouse. So let's get started, and I will turn over to our moderators today, Dragana and Jenny. They are representative of our community, and they will take it from here, and I will see you at the end. I think Dragana is up, right?
Yes. So, hi everyone, my name is Dragana, and I was diagnosed with FA 4 years ago. So, I live in Croatia, and the last 3 years I'm in a group with FA ambassadors. So, thanks for the opportunity to moderate this talk. And for the first speaker, we will hear the Federica Pilotto. So she is from France, Institute Neuro-muscular. So she will tell us more about when the cerebellum loses its rhythm, how FA disrupts brain harmony.
Can I share my PowerPoint? Can you see my screen?
Mm-hmm.
Yeah. Okay, so I'm Federica, and I'm a postdoc in Hélène Pochos lab in Lyon, in France. And I would like to thank FARA for the opportunity to present my bit of my work today. And so let me guide you through this first panel. And so in a healthy brain, the cerebellum, which is depicted here in gray, is like the conductor of an orchestra, where every musician, or in our case, every neuron, is able to play in harmony. And so this allows us basically to move smoothly and in a coordinate manner. In the context of FA, we know that the neurons in the cerebellum are undergoing stress. And so even before the symptoms onset, these neurons start to slowly fall out of synchrony. But the music, let's say, that comes out is still okay. And so somehow the brain is still able to cope with these functional changes. Although with disease progression, unfortunately, symptoms start to arise. And this is the point where every musician basically is playing by its own. And the cerebellum now is totally out of synchrony, and which leads to the appearance of ataxia. And so the imbalance and motor and coordination starts to worsen over time. And so the focus of my research is trying to understand why some neurons slowly start to fall out of tune. And to do so, in the lab, we have a very special microscope, which is called 2-photon microscope, which allows us to do imaging in vivo using animal models of FA, for example, mice. And so with this microscope, we are able to look in the cerebellum of these animals and to monitor the activity of the neurons in the cerebellum when the mouse, for example, is running or is receiving sensory stimuli from the outside or is just simply in a steady state. And in this image, you can see an example of how this looks like. All these dots here, those are different neurons of the cerebellum. And with this technique, we are able to record their activity. And by doing that, what we can see is that during the silent phase of the disease, there are neurons which are, for example, mainly active when the mouse is running or when the mouse is in a steady position. At the same time, there are some neurons which are a bit a mix. They don't really know if they should be, let's say, more active when the mouse is not moving or when the mouse is running or walking. And with disease progression, we see in the second panel that the situation drastically changes. When the mouse becomes ataxic, all the neurons basically now are active no matter what the animal is doing. And so this is the moment where the circuit is basically totally out of synchrony and all the musicians of the orchestra are now playing on their own. And so after this, we can try to identify these neurons that slowly start to be affected by the disease. And here you can see a cartoon depicting the different neurons that are present in the cerebellar cortex. And once we identify these neurons, we can try to basically target them with a specific functional therapy, which aims to basically restore their activity and to maintain their integration in the circuit functional over time. And finally, we have animal models, as I said at the beginning, in the lab. And we are going to try this treatment and then test our animal for behavioral test, which allows us to basically measure the motor coordination and the movements of these animals. And we hope to restore their activity and their coordination over disease progression. Thank you for your attention, and I'm happy to take your questions.
Okay, so we have first questions. Which treatment are you testing? And thank you, says Veronica.
Okay. So for now, we don't have any specific treatment, but we are trying to um, then, once I identify the neurons that are not properly functioning, to use, for example, viruses to specifically target these neurons and try to make them work correctly.
Okay. And the other question, thank you for your research. The alloy to orchestra is very helpful. In picture second, you show different neurons that are active during running and steady. Are these the same as in wild type mice?
So the picture of an FA mouse cerebellum and the one of a wild type is already different before the symptoms onset. But let's say there are some similarities that are maintained, and we are trying to focus on the few neurons that are actually trying, let's say, are already affected before the symptoms get worse.
Okay, so, and another question. Excellent. Thanks, Federica. Which mouse model will you be using? Multiple or?
So in the lab, we have different FA models. And the one that I'm currently using is the one that has been developed from the lab of Marek Napierala, which is the G127B mouse. And we also have the frataxin parvalbumin conditional knockout animal that is it was generated in our lab by Helen Puccio.
Okay, so another one. Thank you so much. What attracted you to study FA?
So I started during my PhD, where I was studying spinocerebellar ataxia. And let's say I fell in love with the cerebellum because it's a very beautiful structure from a neuroscience point of view. And FA is, I think it's a disease where there is a lot to do with this part of the brain, and I hope that this will contribute somehow to get a cure at some point.
Okay, another one. Thank you for your time. If I am understanding correctly, only one neurons are affected. Do we know why? Or maybe I'm wrong.
Well, actually, it's not only one type of neurons, but it's different type of neurons that slowly start to be affected. And so we are trying to understand who comes first. And if we are able to restore this population, this maybe might help also the other ones that are directly connected to it.
Okay, thank you. And the third group of neurons, you do not know yet why they are just sitting there. What is the course of investigation to know why they don't do anything? Thank you for your FA research efforts.
So we don't know yet why they are just sitting there. We know they are doing something that is not, let's say, this is not what they are supposed to do in that moment. And in this way, we are trying to now find a way to push them to do something during a specific type of behavior. For example, when the animal is running or when the animal is not supposed to run. Thank you.
No further questions. Okay, so thank you, Federica.
Thank you.
That was all from the questions for you. So our next speaker is Francisco Figueroa from University of California, Davis. He will tell us more about how gender and treatments shape the heartbeat in Friedreich's ataxia.
Can you see my screen?
Yes.
Yep.
So, hey everybody, my name is Francisco Figueroa. I am a PhD candidate at the University of California, Davis, and I'm working in the laboratory of Dr. Elena Didkova, where we study the heart in Friedreich's ataxia. Today, I just want to talk to you about how we're using mouse models to better understand how Friedreich's ataxia affects the heart differently in both males and females, and how treatment is helping. So first, let's start with this panel. What you're seeing here is a side-by-side comparison of human and mouse heartbeats. These are ECG waveforms. ECGs are electrocardiograms, and they represent electrical signals that control each heartbeat. What's fascinating here is how similar they are. Even though a mouse heart beats much faster than ours, the basic pattern, the peaks, the valleys, are nearly identical. That means that we can study the mouse heart to understand things that we might not yet be able to test directly in people with Friedreich's ataxia. Therefore, this mouse is a powerful model for learning. So jumping here in this panel, we zoom in closer. You're looking at ECG waveforms from 3 different types of mice. Healthy controls in black, Friedreich's ataxia mice in red, and Friedreich's ataxia mice treated with omaveloxolone in green. Here's where things start to shift, especially in the males. In healthy mice, there's a distinct little bump in the middle of each waveform called the J wave. You can think of it like a lyrical verse in a song. It might not be the loudest part, like the peak next to it, but it keeps the melody grounded and cues the orchestra to stay in sync. When that verse is missing, the rhythm feels off, like the song is skipping a beat. What we found is that male FA mice lose this J wave entirely. That missing signal is a warning that the heart's rhythm is becoming unstable. Female mice tend to keep it, suggesting that they have some natural protection in the way that their hearts manage stress. When we treat male FA mice with omaveloxolone, the J wave comes back. That means that the heart's electrical system is regaining its ability to reset and prepare for the next beat. Now shifting over to this panel on the right, we're looking further at the differences between males and females. We're looking at something deeper inside of the heart, the scar tissue, or in other words, fibrosis, seen here in the dark red. Scar tissue acts like speed bumps for the heart's electrical signals. It makes the muscle stiffer and less responsive, and it can also block or slow down the beat. What we found is that male FA mice, or Friedreich's ataxia mice, develop more scar tissue than females. This helps explain why their heart rhythms are more disturbed, and unfortunately, omaveloxolone doesn't actually reverse the scarring. It helps with the electrical reset, like we see here with the J wave, but it does not improve the structural damage. That's something that we'll need to address in addition with other therapies in the future. Finally, in this panel, we use something called a Poincare plot. It's a simple, but it's also a very powerful way to visualize heart rhythm. It measures the variation between each heartbeat. Seen here is this red arrow between 2 large peaks in the ECG. These variations are plotted on a scatter plot, and each dot is a single beat. The shape of the cluster tells us whether the heart is beating steadily or erratically. In healthy mice, the dots form a tight, centered cluster, which is a sign of a stable, predictable beat. In unhealthy male FA mice, the plot shows multiple scattered clusters, which is a visual sign that the heartbeat is jumping between different patterns. That's what unstable rhythm looks like. It is very chaotic. Female FA or Friedreich's ataxia mice are still affected, but they show less disruption. The Poincare plot becomes more centralized, which is reflecting a heart that's holding onto a steadier rhythm. With omaveloxolone treatment, we see a return to a tight clusters in both females and in males, which is good. It's focused. It's more like the healthy pattern that we want to see. However, these clusters are still shifting either to the left or to the right, indicating that these mice are experiencing tachycardic or bradycardic type arrhythmias. So closing thoughts, what does all of this mean? Even though the mouse and human heart are different in size and shape, they speak the same electrical language, and that lets us learn from them. We know that FA male mice heart lose an important electrical signal, the J wave, while females tend to preserve it. Scar tissue is worse in males, and that may explain their greater risk for rhythm problems. Omaveloxolone helps to restore the electrical rhythm, even though it doesn't actually reverse the scarring. And with tools like the Poincaré plot, we can literally see the difference of treatment makes dot by dot. So understanding how these heart rhythms change gives us insight into how Friedreich's ataxia affects the heart and how we might be able to intervene earlier and more effectively in the future. And I'll take your questions now.
Okay, thank you. So we have questions. I did not get what the drug was used to improve the heart rhythm and if this is used in patients with FA.
The drug that we were using was the one that was recently approved by the FDA, the omaveloxolone. It's also called Skylaris, and it is currently used in patients with FA.
Okay. So thanks for sharing your work, Francisco. Is the origin of fibrosis in the heart from muscle cell death or fibroblast activation?
It could be from fibroblast activation, and that the cells themselves are dying in the hearts of Friedreich's ataxia, the cells types.
Okay.
Hi, Dragana. Can I add a clarifying point? Thank you, Francisco, so much for this research. It's really interesting. I do just want to add that OMAV is being used in patients with FA. It's called Skyclarys, as Francisco mentioned, but we don't really know what's happening in the heart of people with FA who are taking OMAV because that was not studied during the clinical trials. So this is really interesting research to start to learn more about that.
Yeah, that's a really good point, and that's one of the main reasons our lab is investigating omaveloxolone with such depth. So thank you for chiming in. Do you know if these?
Okay, I have one more question. Francisco, thank you for your research. Do you know if these same EKG changes are observed in people with FA?
Yes. So the J wave itself is only observed in mice. This is not a feature of an ECG that you would see in a human. If it is there, it's hidden by other waveforms on the ECG. However, we do see an extended QT interval, which is implying that there's a delay in the way that the left ventricle is beating, and other parameters like the P wave is extended, similarly in mice as is human.
Thank you for your presentation. Was there a finding that surprised you the most and why?
I guess, and this is part of our ongoing future research, we're really interested in looking at the autonomic nervous system's effect on the heart, because this might play a huge role in the way that the heart is beating and how the heart rhythm changes. So was I surprised? Yes, I'm always surprised to find out new data, but I'm happy to help where I can and dig into it a little bit closer.
Can you explain to us, Francisco, what the autonomic nervous system is?
Yes. So
just briefly.
Very briefly, very simply, it is the conversation that your brain has with your heart. And what we want to do is be able to understand how the autonomic nervous system is communicating from brain to heart and whether there are ways to fix it. And we really want to see whether our treatments like omaveloxolone is actually helping with the autonomic nervous system itself.
Okay, thank you for your presentation and your work. Do you plan to raise similar tests with other medications, molecules?
Yes, absolutely. We're testing not only omaveloxolone, but we're testing a multiple set of drugs at our laboratory, in addition to combination therapies.
Okay, another question is why the difference between sexes?
Yeah, I feel like this is a contested argument. Like, I know that Friedreich's ataxia affects both males and females differently depending on the GAA repeat length. However, it's already been reported by our lab that we've seen a sexual dimorphism in these mouse models, But in addition, we do see reports about cardiac differences between males and females, published by Dr. Schatt or other people in other laboratories. So there are sex differences when it comes to cardiac muscle that we should be aware of.
Okay, so I don't have any more questions for now. It should mean that thank you, Francisco, for your presentation, and I'm leaving you to other moderator. Thank you.
Okay, so that would mean that it's my turn to introduce myself. My name is Jenny Holcomb, and I'm part of the FA community. I started being involved in late 2018, early 2019, as I was on the diagnosis journey with my son. And we were diagnosed in April of 2019. He just turned 18. So we're still on the journey, not in diagnosis anymore, but very interested in all of the research and all of the wonderful things that are going on behind the scenes that we get to hear about. So our next presenter is Carla Pernacci, and Carla is going to, she's from University of California, San Diego. She's going to talk to us about targeting microglia to fight neurodegeneration in FA.
Thank you for the introduction. I'm going to share my screen. Hi, everybody. I'm Carla, and I'm a postdoc from Dr. Koppel lab at the University of San Diego. I'm working on a unique cell type of the brain, which is called microglia. Those are the immune defensor of the brain. To study this cell type, we take advantage of human-induced pluripotent stem cells that are derived from FARA patients and healthy donors. In our FARA iPSCs line, we also apply CRISPR-Cas gene editing approach in order to correct the abnormal trinucleotide repeat expansion in the frataxin gene and to restore frataxin level in microglia to investigate the role of restoring frataxin level in microglia cells. Microglia here in this in the blue panel here, at their homeostasis condition, they show ramified morphology, and they release anti-inflammatory cytokines as well as a growth factor that sustain neuronal survival. However, in different neurological conditions, those cell types became hyperactive. When these cells are pathologically active, they became round in morphology and therefore amoeboid. And when they are in this state, they start to release toxic factors, including pro-inflammatory cytokines, reactive oxygen species that are harmful for the brain. The release of this factor also contributed to the degeneration of neurons. In the context of FARA pathology, neuronal degeneration has been shown specifically in the cerebellum, where the deep cerebellar nuclei tends to be more prone to frataxin loss. And more recently, microglia involved in FARA pathology has also been documented in mouse model mimicking FARA pathology. In our lab, we first wanted to confirm that microglia are involved in the pathology using human sample. So here we used postmortem human tissue from FARA patient and healthy donors, and we looked at cerebellar cortex of those patients, and we stained our section for EVA1 in order to understand microglia pathology. Here we are showing that FARA patients show an amoeboid and more round microglia morphology, which is indicative of a pathological activational state of these cells. In order to understand the pathological consequences of frataxin loss specifically in this cell type, and to investigate whether FARA microglia can somehow influence neuronal survival, we use a healthy mouse model, which means that all neurons and all cell type of the brain express homeostatic level of frataxin protein. And we transplanted in those mice FARA microglia derived from patient, as well as microglia derived from healthy donor and gene editing lines to understand how FARA microglia and if FARA microglia could interfere with neuronal survival. At first, again, we looked at microglia morphology, and we again observed that this amoeboid and more round FARA microglia morphology compared to healthy donor microglia. And importantly, we also showed that the gene editing approach also restored this homeostatic morphology of this cell type. We then looked at Purkinje neurons, those are cerebellar neurons, and by counting the number of those neurons, we showed that the mice, the healthy mice injected with disease microglia, show a strong neuronal degeneration, which is specific for Purkinje cells. And again, our gene editing approach, specifically in microglia, was able to sustain neuronal survival. So what we are doing now, we are investigating the therapeutic potential of our gene editing approach in microglia to dampen neurodegeneration in FARA patient line. Happy to take any questions.
Okay, thank you very much, Carla. We'll wait on a couple of questions to come in. Monitor the box. I'm curious, while we're waiting, about the cells. You've done the editing, you've gone back to the mice, you've counted the neurons that have the degenerative properties at that point. That seems to be effective. Are there any observational studies of the mice in terms of their activity or their behavior after that happens?
We are working on that. We are now investigating why those healthy neurons degenerate as a consequence of disease microglia addition to the brain, as well as the mechanism driving microglia to be hungry to the point to attack healthy neurons.
All right. So I guess any visible impacts to the activity of the mice, though, after that editing, after those edited, so stuff has been added back in, and then is there anything that's changing about their behavior at that point?
We are working on behavior, but I am collecting right now survival data. So what we are actually looking at is if we are able to prolong the survival of the mice after gene editing in microglia.
Okay, wonderful, thank you. We have a couple of questions. One, how can, I'm not sure of the word, how can such gene editing be made with humans? What type of steps are?
So we do have a dual RNA guide that it's able to recognize the sites where the GAA trinucleotide repeats start, and it's able to make a very precise cut to remove the abnormal trinucleotide expansion. So those dual guide RNA are right now passing the quality control to then be applied as a clinical trial.
Right. Let's see, Carla, thank you for your work. Did I understand you correctly at the end? Are your next experiments to give healthy microglia to FA mice to see if the healthy microglia can have a treatment effect?
Exactly. So what we are doing right now, we are firstly differentiated neurons using the same patient line that we have been using to characterize microglia. But this time we are differentiating neurons from FARA patients, and we are co-culturing them with gene edited and therefore FARA corrected microglia to see firstly if in the dish we are able to sustain a neuronal survival. And the next step would be to cross our FARA mouse model with this healthy mouse that actually has a complete lack of endogenous microglia, where we will be transplanting these gene-corrected microglia and look at FARA neuronal survival from a mouse model.
Wonderful. Okay, so the next question, thank you so very much for your FA research. What will be your next steps, and what are you hoping to find out?
I'm really hoping that this gene editing approach will pass the quality control and go ahead with the clinical trial, because we already have data that we published about FARA neurons that upon gene editing, they prolong their survival in vitro. So we really hope that we could provide a therapeutical approach and focusing on microglia, which is a new cell type that has been discovered in FARA pathology, could definitely help in understanding the cell type specific contribution of FARA pathology.
Fantastic.
I have a quick question. I know you touched on it a little bit, but do you have an idea of why the microglia is activated in FA? What causes the microglia to get activated?
So what we know from our side is that lack of frataxin specifically in microglia not only recapitulates some hallmark features of FARA pathology, like, for example, the activation of reactive oxygen species, for instance, as well as mitochondrial accumulation and dysfunctional lysosomes. All of these dysfunctional organelles basically build up within microglia that is not able to clean all of these debris that start to accumulate within the cells, and these might influence the release of these toxic and dysfunctional organelles that will be possibly picked up from neurons.
Okay. I don't see any other open questions right now. So thank you. Thank you very much,
Thank
Carla.
you. Thank you.
We will move on to our last presenter. Hemin Cho is from, lost my notes, Stanford University School of Medicine, and he will be talking about myeloid cell replacement therapy to alleviate symptoms of FA by intercellular mitochondrial transfer. Thank you, Hemin.
Yeah. Start sharing. Okay. Yep. My screen is shared.
Yeah, we can see it.
Yeah. Hello, my name is Hymin Cho from Stanford University. Today, I'd like to share new therapeutic approaches we are exploring for Friedreich ataxia, FA. One of that aims to restore brain cell function by enabling mitochondrial transfer between cells. We begin with one question. What if we could supply FA brain cells with healthy mitochondria? Could it help we can be recovered? To restore this, we used an FA mouse model. We first performed a process called conditioning, which can remove the disease microglia from the brain. Then, for this one, we involved two drugs. For the busulfan, to reduce the disease cells, and PRX3397 to suppress certain immune cells and allow them to brain engraftment. And from the mouse model, we can see like this one about the well engraftment of the mouse brain cells. Next, we transplanted the healthy brain donor cells. Then these cells migrated through brain and replaced it with microglia. These new replaced microglia, they deliver their own healthy mitochondria to nearby damaged cells, including neurons, astrocyte, and oligodendrocyte. As a result, we observed very clear sign of the cellular recovery, increased the ATP production, improved the oxidative phosphorylation, and reduced the stress markers. Importantly, the treated mice are also showing better behavior tests and improve the overall health status. So this therapy is not just for replacing of cells, it is also rebuilding the supportive network in the brain throughout the intercellular mitochondrial transfer. We believe this kind of the mitochondria-based support from healthy microglia could represent new direction for FA and other neurodegenerative diseases with as well. Thank you. Yeah.
Thank you, Human. We will wait on some questions to come in. This seems like quite exciting research, very similar in some ways to Carla's. There are definitely some parallels in the things that you're doing. Is that correct?
Yes.
Thank you, Carla.
Can I ask one question? You talked about maybe you can explain a little bit how this treatment would work in people, how you'd actually, what the process would be like,
Sure.
where do you get the cells? How do you? But I also was curious, and maybe you can explain while you explain the process, why you need this conditioning
Yes.
regimen.
Sure, absolutely. I can starting from the conditioning and what is difference with other cell type. First of all, I will sharing the screen. Yep, I will share with this screen. First of all, to understand this process, we should notice what we are using for the donor cells. We are using donor cells for, we call it the positive cells. This mice has genetically modified it. They express the whole cells GFP, and their mitochondria express the MK2. So when we transplanted these cells, we can following the donor, where donor cells going, and also following the mitochondria transfer is happening or not. So in case after transplantation, if in the brain, well-transplanted cells, we can check with the GFP cells. And in case of the mitochondria transfer, can you show this one well? Okay. In case of the mitochondria transfer, we can check from the donor cells with the positive and recipient cell that only express the MK2 positive cell that take the mitochondria. So from these two cell types, we can follow the mitochondria transfer between the two cell types. And second thing to explaining why we should do or why we can try to about this conditioning is it's recently that publication is recently news in case of Parkinson's disease therapy. The Kyoto University and the Blue Rock therapists, they try to inject from the iPS delight neuron cells. But in that case, we need two things, like we should inject the cells directly by injecting the needles in the brain. But if we're using this conditioning as Carla introduced, FIDA status of microglia is already disease or very patient status of the microglia. But if we are using this conditioning-based method with the busulfan and PRX to combining method, we can transplant it or we can deliver it healthy donor cells in the brain without any surgery processes. Yeah.
Thank you very much for that answer. There are a few other questions. Did you look for mitochondria transfer just in the microglia or brain or other cell types or organs?
You means in the in vivo status or in vitro status? Because we also comparing two things.
The question was not specific to that. Sorry, Liz.
Yeah. No, sorry. I was gonna say that maybe to that, maybe the in the mouse and we talked a lot about the microglia in the brain, but what about other organs?
Yeah. So in case of today, I just delivering the information only for the brain it's just a 5 minutes talk, so I don't have time to explain all the tissues. But in our papers, we are showing starting from the brain, and we did the total bone marrow transplantation. So we also did total bone marrow cells, and in these tissues, they are also showing mitochondrial transfer, and it's our in vitro experiment. And if we do the co-culture with the double positive macrophages, derive the macrophages and fibroblast of FA status and wild type status of fibroblast, we can confirming mitochondria is transferred between two cell types. And also we also confirming in the heart. I don't have the data in here, but we also confirming the mitochondrial transfer in the heart. But in case of heart, we have the little difficulty to analyze which cell types mainly take the mitochondria. Is when we want to analyze by the microscope, the heart has their autofluorescence is very high, so we cannot detect the MK2 signal well in the heart. And in case of heart, the cardiomyocyte is the size is very big, and their tissue is very difficult to dissociate. So from the heart tissues, I cannot detect the flow analysis, how we can detect the mitochondrial recipient cell types. But except the heart, we are checking spleen, peripheral blood, and bone marrow, and the brain. We check the mitochondrial transfer in these tissue types.
Okay, so there's another question. Is there a preference of the replacement microglia for one area of the brain versus another?
So you mean in this?
Well, I think,
yeah.
I was just going to say, I think you mentioned that it would be good to be able to directly inject the microglia into the brain, but instead there needs to be a transfer. If you were going to inject, I suppose, would a different area of the brain be better than one versus another?
Yeah, I also think instead of injection of the one location, we already know FA cases, the cerebrum is the most significant location for microglia or disease status. But I think that the distribute all of the microglia became replaced in the whole brain. I think that can be more therapeutic or more helpful.
Okay. Another question, could the added microglia be affected by inflammation within the FA brain?
From these mice, we cannot detect any inflammation. But because absolutely the PRX status, PRX 3397 can reduce about some can reduce can suppress some immune cells. So I also a little worried about this part. But from this mouse result, I can tell there is not significant inflammatory or any symptoms of that. And we just told it becomes recovered.
Okay. Let's see, one more. How do you distinguish between healthy and unhealthy mitochondria following the transfer beside seahorse test, sorry.
Seahorse test, okay.
You mean as a slide for every question?
Yeah, I'm sharing all but we already submitted this paper, so it's fine. But we did the seahorse experiment like this. After culture of the double positive and FA disease fibroblasts, and we detached FA monoculture and wild type status monoculture, and from the co-culture, we can, from using the flow sorting, we can sort it FA cells without the mitochondria after co-culture, and we can sort it FA cells with mitochondria co-culture. So from these 4 different cell types after detachment and counting the same number of cells and plating in the seahorse plate, and we check the seahorse experiment with the 4 cell types. And from here, we can completely to see wild type status and FA status. They showing big difference in seahorse experiment, but without mitochondria transferring, there was not big significant enhanced in FA cells. But after taking the mitochondria, they see the big differences after co-culture. And this method, we are using directed co-culture. And one additional thing is, one of the reviewer asking us about how to check about the indirect method. Because we are just, in this story, we are just talking about mitochondrial transfer by just direct co-culture, cell to cell, in direct co-culture with nanotubules. But for revision experiment, we also expected whatever happens with the indirect co-culture system.
Thank you very much, Eamon.
I think there's Oh,
is there
sorry.
another
I
question?
see one more question from Mary. What findings surprised you the most, or what's your favorite finding? Is there anything that was surprising when you did these experiments? Like, were you expecting the mitochondria transfer, for example?
Actually, before we check what happens or what we did, what is the mechanism due to the single cell, but primarily most surprise is the phenotype of the cell. This is mice recovered. After we did this therapy, we started, okay, let's do more deeply analysis for this one, because as you see this status of mice is not fully, but it's symptom the body weight or behavior is significantly enhanced. So I was really interested from here. And the second thing is, when I'm the postdoc now, but when I do the PhD degree, I have not assessed with single cell or any information data. But from here, when we detected that NK2 positive cell types that take the mitochondria, and if we are checking from all cell types, they take the mitochondria, or they recover the mitochondrial respiratory chain complexes are recovered, that part was surprising for me. Because this part, I think FA was most affected with this chain system.
Okay, I think that's all the questions that we had.
Yeah.
Thank you again, Eamon.
Sorry, I just expected just 5 minutes of talking, but I brought 25.
That's good. All right. Well, I would like to thank the speakers today. They were all fantastic. I really enjoyed everybody's talk, and everybody did a wonderful job. And thank you, Dragana and Jenny, for moderating. We're really grateful for taking on this job. And thank you all for listening. We hope you enjoyed learning about some of the research that FARA is funding. And we'll see you in 3 weeks on May 28th for the more flash talks from junior investigators. Have a good day. Take care, everybody.
Thank you.
Thank you. Bye.